So, welcome everybody to our meeting. So today I'll talk about the introduction to single-cell perturbation Kaggle challenge. So the talk maybe will be most be introductory and educational and intended for those who have not yet started. And so I'll describe the task, metric, some baseline, some public solution, cross-validation schemes, and some kind of more advanced approaches like target encoding and so on and so forth. Okay, so just a kind of information. So we have this kind of meetings quite regularly. In particular, we had some meetings like that for the previous Kaggle competition. And if you go to our YouTube channel and the playlist in English, you can find some videos which were related to the last year and even this about the 2 years ago solution of the open problems competition. So here are some, this one by Tiano Lee is one of the winners, and the other some introductory talks by biologists and so on. And we have some videos related to the other Kaggle competitions. Yeah, so you are welcome to subscribe to our YouTube channel and Twitter and Telegram if someone uses. Okay, so let me start. Describe the task and the day. Sorry. Okay, so just the main thing, goal, what we need to predict. Just in the simple words, we need to predict the effect of the drugs under the cells, for action of the drugs on the cells. Yes. So organizers have created the data set where they treated about 146 drugs for 6 cell types. And we have the data which show us, quantifies the effect of the drug on these cells. So more precisely, we have the quantification of the effect of the drug on about 18,211 genes. For those who are not biologists, I'll explain later what genes and expressions are, but for the moment, it might be not important. So some bigger context for this kind of tasks. So it's a kind of quite hot research topic. So in general, we want to find the effect of the different chemicals, different compounds on different type of the cell types, on different cells. Ideally, someone gives you the chemical formula and you should say what is the kind of effect on these cells. Of course, this is a kind of, we are quite far from this goal, but researchers are trying to do this. Yes, and so if you look into more detail, so in the train, we have only about 600 samples, and in the test, we have only 255 samples. Yeah. So from the data science point of view, this competition is maybe not that much kind of exciting because now the sizes of the samples are very low. And that means we cannot build some big models, and we should be afraid of the shake-up because there is big amount of the variation, which will be quite random for such a small sizes. Yeah. So a few more mods for the people with some biological background. First, what I want to mention, despite the competition is called single cell, the actual data which we have, which we need to predict, is so-called pseudo-bulk. That means we have averaged all the single cell expression for the certain cell type. Yeah. So for data scientists, maybe it's not important to understand what's going on, but just for biologists. Yeah. So it's a not prediction for each cell, but only 6 cell types. only 6 cell types. Again, for biologists, so these are the cells, the healthy human blood cells, T cells, Trx, CD4, and CD8, plus and myeloid and natural killers. So they're from human healthy donors and 3 different donors. And the treatment was for 24 hours. Just some names of the drugs for I don't know who knows, Verinostat, clotrimazole, taptican. So there are many kinase inhibitors, and there are 2 controls. One is negative control, DMCO, which is solvent, and positive control, it's belinostat and darfinib. So these are the drugs for which we know they have a very strong effect. So they selected as a positive control.
So can I ask a question? Sorry for interrupting.
Yeah, sure.
This means that all of the drugs that are used for testing, they are candidate drugs that can mimic the kinase inhibition property, or what exactly are we trying to get from those drugs?
Not all of them. I think about 15, something like kinase inhibitors, but there are many drugs. For example, my friend said that clotrimazole is something like protivgripkovый. It's anti-yeast, something like
Antimicrobial, no,
Antifungal.
yeah,
Antifungal.
antifungal.
So
There was also antidepressant, I saw.
Antidepressant, well, okay, so there are more than 140 drugs, so I don't know all of them, but just...
Yeah, my question is more about the belinostat and dabrafenib, if those have some specific action. Why are they called positive control? How exactly they affect all those cell types that they are called positive control?
What I have read in the forum discussion, the ideas was the following. So these drugs are adopted by FDA. They're quite well known, and they know that they have a strong effect on the cells. So the organizers select them as a kind of positive control. That means if they see that for some cells, these drugs doesn't have an effect, that probably means that something is wrong with this kind of plate or maybe cluster of the cells, and you should throw it out. So as far as I understand, the idea was something like that. That, I mean, so in simple words, these drugs should have a strong effect. If you don't see it, then something is wrong. It's something you should kind of... Is it clear?
Yes, thank you.
Okay. Yes, so organizers have some comments. So this compound was selected based on the diverse transcriptional signatures observed on this type of the cells, whatever it means. Yeah, I mean, okay, it's clear. Yeah. If someone have a question, please ask. So, main goal is to predict the effect of the drug. This, I think, important to understand. Yeah.
So do you have any sense what does it mean by diverse transcriptional signatures?
My guess is that they looked on the changes of the genes, and they wanted to select not so many drugs, but that they change many genes, many kind of one drug is changing one thing, another drug is changing the other thing, so kind of get a diversity of their do you understand? Yeah.
I see. Thank you.
Yeah. So transcriptional signatures, that means the transcription, that means expression of the gene. So when you say signatures, that means not one gene, but maybe many genes. So when you say transcriptional signature, that means that kind of this drug, for example, changes that particular 100 of the genes. Another drug changes another 100 of the genes. And you want to select not so many drugs, but such that they cover as many kind of diverse genes as possible. You cannot select. So, of course, I think we want this experiment to made for many drugs, but it is very expensive. So the restrictions that we have only 140 drugs, that's because this experiment is very expensive, and it's kind of course, everybody wants to test all the drugs, but it's impossible.
Yeah, but it's make a question of how would be generalizability of this competition to many more drugs. Like these results we get will work on just only for this kind of drug, which produce diverse transcription signature, not for all.
Yes, yes, but let's go to the next slide. So that means that yeah, so it's answering your question in some sense. So what we have here, important thing which we should understand, I think, is the following. So the drugs in the same in the train and in the test are the same, and the cell types, we have the same cell types in the train set and in the test set. That might be unusual because for many kind of research in this topic, people want to generalize the effect of the drug on something unseen drug, or some new drug which you do not have in your train set. For many researchers, task is like that. But here, the task is different. Here, the task is that we want to predict the effect of the drug on the different cell type. That's the main point of this competition. So you see this picture. So this dark blue, this blue is a train set. And this light blue is a test set. And here below are the name of the drugs. And so you see we have information in the train for these 4 cell types, or you can see the same magnified here, and we need to predict it for the other cell types, for the same drug. So for this drug, we have this information for these cell types, and we need to predict for this cell type. Yes, so the point of this competition is to be able to generalize to the different cell type. Does it make sense? Antonina?
It does make sense from the computational standpoint, but does it make sense from the biological standpoint? That's the question, because it's not necessarily given that all drugs of those selection of those sample will have similar effects on those cell types. Although obviously you need to look into the specifics of each drug, but still it seems a bit strange that they assume that all of those drugs will affect different cells like in the same manner, even though they obviously do not mean that they have to affect exactly B cells and myeloid cells exactly as the 4 other types, but that sort of the logic of this drug affecting those 2 cell types will be predictable.
But just, and so there are some details. For some drugs, we have full information. This part, it's about 15 drugs. We have information on them for all the cell types. And the point is that, for example, this drug is a kinase inhibitor, CDK2 inhibitor, yes? So this drug is similar to some of these drugs, yes? And if you know its effect on these cell types, on B cells, then you can kind of predict the effect of the similar drug, yes? So in this competition, it's not that you completely do not have information of the drug on these cell types. Exactly. Additional information because some of the drugs are similar to those which are given to you.
Exactly, this I understand. Like my concern is that in the world of chemistry and its interaction with biology, it is often hard to predict each minuscule interaction that can occur, can be raised by a very small change in the chemical formula. So it is very hard to predict how addition even of a small additional chemical group will affect some tiny details. Like you obviously can predict a lot of general chemical properties, and you can predict like the way your drug will interact with a broad class of proteins, but it is possible that some individual interactions with the drug and the concrete protein will occur, and it's very hard to predict those things, and they can have a very high effect, I think. But yeah, so in essence, my question is that you probably want to look at the chemicals as close as possible and understand how exactly they, like what kind of clusters they form, and if there are some quite like distinct chemicals from the rest, or if there are some clusters in which we have a whole family of chemicals that are very similar and that are known to behave similar. Anyway.
Well, it's a good question. Also in the chat, Hrnbek asked that similar question, how can I find information what drugs are related? So the first thing, we can look on the effect on these cells. So we can just, so we have for all the drugs, their effect on these 4 cell types. And we can cluster the drugs based on this kind of effects on these 4 cell types. And it will give us some information. This is one of the approaches. Another approach is just to look into more chemical things, chemical stuff. And I hope Antonina will present a little bit about the molecular descriptors. It's a kind of way to work with these chemical formulas. And another way, for example, these embeddings by Cambert, which was shared by Alexei Trepetsky, we also can find the embeddings, then just cluster the embeddings, and we will get what drugs are related. Yeah, and there can be many other ways. For example, very naive way, you can see that some of the names, some of the drugs, they ends IB. Yeah, that means something like inhibitor. And there is some, it's not very precise, this naming a little bit kind of, well, not well, the naming procedure is not well defined, but still they give some information. You can, for example, if it's CLIP, it seems it's like something like CDK inhibitor and so on. So by the ending already gives you some information what kind of the drug is. Of course, this is a kind of very, not very precise, but something. Yeah. So EB at the end is inhibitor, and you can find this in Wikipedia. It's easy to kind of Google this. Okay, makes sense.
Yeah, there are pharmacological groups actually which divides these compounds based on their action, mechanism of action. So you can use this or some else classification. There's a lot actually.
Yeah.
How to divide drugs into groups.
Yeah.
A lot of work.
Yeah, but a kind of first approximation here we can get just from the name, but of course this is very vague. So, yeah. Okay. So let me now give some information. Yeah, show just the train data. Yes, how the train data and test data it looks like. Yeah. So just we have this. If we plot this file with the train data, we will see the first column is cell type. The second column is the name of the drug. This is ID of the drug, and this is chemical formula. Yeah. Now it's a control or not. And then then there are these 18,000 columns which correspond to genes, which are denoted typically by this kind of symbols. Typically 4 symbols. It's the names of the different genes. Yeah. So in the test data which we need to submit, we have only 2 columns. That is cell type and the name of the drug. So basically we need to, we have only 2 features. And we need to predict only for these B cells and the drug name. Yeah. This is the sample submission, which is filled by zeros. So its size is 255 by 18,211. Yeah. Is it clear? So I think those who already started know this quite well, but maybe someone haven't yet started, and it might be something interesting. Okay, so the next thing. So so from the data scientist point of view, I think we can skip this slide because we have some data in the train set and we need to predict on the test set. And we don't care much about what this data is, yes? But if we go to the more biological sense and if we'll go into the details, then these details will be very kind of difficult. Yeah, and let me just say a few words about this because it might be useful for further questions. So yeah, so what are these numbers which we are predicting means? First, this is effect of the drug, but effect comparing with the control, with the negative control. Yes, so and this negative control and the effect of the drug are calculated for the same cell type. That means you have some cell type, for example, myeloid cells. You have some negative control, so that means nothing happens, and calculate the expressions of the genes for this negative control. And then you calculate for effect of the drug. And then you compare the difference between the negative control for this particular cell type and for effect of the drug on this particular cell type. Yes. So we never make a comparison between different cell types in this task. That's one of the points I think which should be clear. It may be important. So so these numbers which we have, the effect of the drug relatively to the control and control in the same cell type. The other thing, also quite simple, that these numbers are calculated not for single cell, but for average of the thousands and thousands cells of the same cell type. Yeah, this means called pseudo bulk. Yeah. So
and raw values are not available.
they're available. Single cell values are available for train, not for test, of course.
But these are not significant p-value. We have only, I guess, counts, normalized counts, but not p-values or something, right? But at the level of cell type, of cells, the level of cell. Level.
The p-values are calculated on the level of these cell types, not for cells.
Mm-hmm. And on the level of cells, we have also counts, right?
Yes, yes, we have counts, and moreover, we have ATAC data for train, not for test, of course. Okay, so I think these two comments about the might be more or less simple and maybe clear, but now these comments, these parts 2 and 3 are more kind of delicate. So, so first, when we quantify this effect of the drug, we take p-values. We don't take the log-fold change, we don't take the mean change, but we take these p-values, or more precisely, logarithm of the p-values, and multiply it by the sign. Yeah, and these p-values are calculated by the standard bioinformatics package lemma. Yeah, and so on the meeting there was organized by the organizers of the competition. So it was a discussion why we take these p-values. Yes, why we take lemma. there is no consensus in the research community that this is the best way. There are many packages. Sometimes people say, you know, this package is good, another good. So there is a kind of not good answers for these questions. Yes, but we have what we have. Yes, so we organizers have chosen lemma and the p-values.
One question. In the test set, am I right that they will they use the same controls or different controls? Because
control is always, as I say, this DMCO, which is a solvent.
No, I mean, if what we are predicting is a differential signal, the differential signal comes from a comparison between the potentially perturbed cell and a set of control cells, right? So
control cells are the same cell type, so they will be different.
So in other words, we do not know, we do not have any differential expression profiles for even cells of different for the same cell types. So I don't know. I think my question is more or less clear. Like the question is whether in the test set the differential values, those p-values, are against the same control cells or not. So you say that they are against different control cells.
Yeah, because you make, as far as I understand, comparison for the same cell type. You take B cells which are in the test. You do not have, I mean, and compare the B cells without drug and with drug, and that numbers are given to you. Yes. So here, all these numbers is comparing T cells without drug and T cells with drug. Yeah.
Yeah, like what I'm confused about is that you can see on this picture that there are values in the trained set for B cells and myeloid cells just for other drugs. And as I understand it, we make a comparison between a cell affected by any drug and the cell affected by a DMCO or those two drugs. So, I mean, it doesn't matter. The control for each drug is the same.
Yeah, the left column is positive control. It's not, it's the other control. It's not the control which we are kind of calculating differential expression. Yes. So the negative control is not shown here, I think.
Yes, but implicitly for the last two rows, for the cells that are marked as trained, implicitly those values are obtained with respect to some control. Maybe this control is not present in the training set, but it is present in the way those values are governed, right? And my question is whether we will whether the holdout set will be the same control cells or not. But anyway, I more or less understood.
I think this kind of raw values for single cells, they are available for this control even for these cells. I think so, but I haven't checked. Yeah, but here it's okay.
Yeah. Okay. Thank you.
Okay. So a little bit comments on what is lemma, what's going on here. It is the following. So again, it's for data science point of view, it's almost unnecessary, but just for biologists. So the cells which we have, they're from 3 different donors, and moreover, they were in a little bit different plates or some conditions of the experiments. And so we don't want to see the variation which comes from the donors or these plates. This is technical. We don't want to see. And so, and so this lemma package tried to build linear model, very simple, it's written here, to remove this effect. that we don't want to see changes from the cells are from different domes. We only want to see the changes which comes from the drug effect. But if you don't do this, if you don't do this removal with lemma, then it might be some of the changes will not from the drug, but just from the fact that the cells are from different people. Yeah. So that's the idea, at least how I understand this. Yeah. Okay. Something is clear, no? Okay. This is easy slide. So in total, we have $100,000 prize for this competition. But what is unusual, it's divided into two parts. So for 50,000 comes for the standard Kaggle procedures, which means if you get the good score on the leaderboard on the private part, then you'll get this money. But there will be 50,000 for the so-called judges' prizes. So as far as I understand, the idea is that they will kind of try to estimate by the experts if your contribution is valuable for the research community for something. And so they give us the question list. Part of the questions is on the slide, but not all of them. Please look on the competition site. So they, for example, ask, that might be interesting for biologists, how do you incorporate the ATAC data? How do you incorporate this links database? How to incorporate chemical structures and so on and so forth? Yes. So it's more kind of to stimulate the researchers to kind of participate and maybe win this prize. I think it's a very good idea, and that we can hope it will stimulate the researchers to do something. Okay, and so we have more. So we can say that competition is at the beginning, only 23 days. It's just beginning. Yeah. Okay. Oops. Now we go to the baseline. The first very simple and very trivial baseline actually doesn't work, but just to demonstrate how the most simple. Yeah. Just all the baseline, it's this lines of the code. Yeah. So we load the data. We use CantBoost regressor, and about CantBoost, we know that you can give CantBoost the categorical features, cell type and drug name, just as it is, without any changes. You just say these features are categorical, and please train me the model. Yeah, and it will train you some model. Actually, it will be very bad, but doesn't matter. Yes. Another important step which everybody is doing in this kind of competition, that everybody is doing the reduction or dimensional reduction of the targets. Might be a little bit unusual because typically we do it for features, but here if you do it for targets. So we take this Y, it's the targets, yes, and we make, for example, truncated SVD, dimensional reduction to 25 components. And then we model is predicting this reduced targets. Yes, we train the model, feed command, then we predict for the submission part, and then we use inverse transform, and we get from the 25 components, we get all this 211 targets. And that's all. We just create the submission data frame. Don't forget to make ID for the index name, save it to the CSV file, and make submission. And that's all. So this is just a simple baseline. Yeah. Is it clear? So the ideas which we have here, what are important? Important is that we always can do the reduction of the targets, because it's very difficult to work if we have this 18,000 targets. It's very difficult to work with them directly. That's why we need to do first some dimensional reduction, predict only these targets, and then make inverse transform. Yeah. This trick with the dimensional reduction is always used in such kind of competitions. Is it clear?
Nice to hear. I actually missed this part and didn't do this. I predicted all 18,000. Should learn how this truncated CVD works, because it's just crazy for me. How it reverse from 25 to 18,000 back.
So no,
That,
truncated
rotation?
SVD is more or less the same as PCA, for example, yes? And how it works with this inverse transform, in a sense, it's kind of fills the other components by zeros, yes? So it's like if you kind of put here all the components, yes, and you make inverse transform, you'll exactly find the initial matrix. But if you make some component zero, no, you can get some approximation to it. And because this is a kind of PCA stuff, first component is incorporating the most of the variation of your data. If you keep only several components, you think that you take the most important information. And you think that other information is a kind of noise which you can neglect. And actually, so we think that we are doing this mainly to speed up the process, because it's to predict this 18,000 targets is quite difficult, yes? But actually, not only for speed, it is useful for quality. It gives a better score, because this reduction, in some sense, it kills some noise. Because we think that smaller components of this PCA or truncated SVD mainly containing the noise, which you're unable to predict, and you don't need to predict the noise. And that's why this trick is always used, and and typically it gives improvement of the model.
So this way you predict only important targets, and all the other gets zeros.
Yes, but you think the others are kind of noise, which you will not be able to predict anyway.
No, they actually do not. That's not the case in the original gene domain.
Yes,
you do predict values for each gene. It's just that they come up from 25 hidden values.
But you make reduction to 25, and then you make inverse transform, which returns you all these 20,000, 18,000 genes, yes?
And always some numbers, right?
But yes, but so even if you my idea is that to keep only main components is good, because the other components may be mostly noise, and you will not able to predict them. And if you will try to predict noise, you will spoil your model.
This like I think that I trust that if for a long time people are doing this in such a way, this is definitely a sensible and like conservative approach. Still, the problem here is that in any dependent sort of cluster, or I forgot how it is called in the SVD, but a sort of a cluster of closely correlated in their expression genes, what you can have is sort of a single gene that is really responsive to your drug, and other genes that are maybe a little bit responsive, but very randomly. And then potentially you can have some, like you can have a situation where the noise from other off targets will sort of reduce the signal for your main gene. So obviously it may be still problematic. Especially if you are working with log p-values, like because log p-values linearly are quite close to each other. But I guess for SVD it's okay. Maybe it's even better. But still, even a small signal here is important, right?
No. Small signal is important if you can predict it, but typically small signal is under the noise is so big that kind of small signals will be lost.
Obviously. So I think that if you
if you will be working with the data set itself, like if the data set will be the main source of the information in your model, then it is super sensible to do it this way with reduced dimensionality of your outputs. However, if instead of working with this dataset, for example, you will create some generative model that works with single cell or pseudo bulk cells, and instead of having only those like 18,000 samples, utilizes like everything you can find, then maybe 25 dimensionality is not that good. Maybe you can expand it, for example.
Yeah, but here we can check everything. We can just reduce it to not 25. I have tried to 100, to 250 different components, and it seems 25 is okay for that particular model. Yeah, so we have the cross-validation score, we have leaderboard score. You just check what is better. Yeah. So here for this task is simple to answer this question. Yeah. So just a little bit for the details. So this is the CatBoost model. As I said, it is just a demonstration. Yeah, the score will be very bad. I mean that for this particular parameters, you see one iteration, depth one, it means it's not learning. The prediction will be just by mean. And if you use these parameters, it's the same as kind of just calculate the means of the so the average of each gene and submit it. It's more or less will be the same. Yeah, and so I tried to optimize by Aptuna on the cross validation, and I tried to optimize only these parameters, and that was the best result. So the other were even worse than just a trivial submission by mean. So I don't think this approach with just using categorical features is very good or promising. Maybe we need to optimize other parameters of the CatBoost, but I don't think it will work. But we can try. Yeah. So okay. So this is a kind of maybe the next slide is a leaderboard. Yes. So you see that the top result, it's appeared today, is something like 0.571. Yeah. Yesterday this result was top. Yeah. And here we have the public best public notebook. It's like this. Yes. And there, so just to give you some impression of what's going on. Yeah. But just to understand good it or not. Yeah. So we need to compare with some kind of trivial baseline. So the trivial baseline, for example, if you submit all the zeros, what will happen? If you submit all the zeros, you will get this score, 666. Yeah. If you will be more clever and submit means or medians, you'll get a little bit better score or percentiles. Yes, that's a trivial solution. For each gene, it will be the same constant. So the constant will be different for genes, for different genes, but it will not depend on the cell type or the compound or the drug. Yeah? So this we consider as a kind of our first benchmark, so kind of trivial baselines. Yeah, these numbers. Now top solution is like this. So not so bad, I would say. Yeah. So that means we are kind of a little bit predicting something. Yeah. So this is a very trivial baseline. The trivial baseline is to do the same, but to make the average median different for each compound. That means you take the train set, you fix the compound, and you calculate the, for example, median or mean or some percentile for all the target values for this compound. That means you average all the cell types. Yeah. And if you make a submission with this, you'll get this kind of score, which is a little bit better. But again, it's trivial baseline. It's even another kind of baseline, which is again no actual learning. It's we can do this denoising, which we discussed before. That means we kind of first do this as TSVD, keep only 35 components, and then go back. And then use this quantity. And you'll see we get a better score. That I was trying to explain before, that this reduction of the components by TSVD is helpful, despite it seems to be kind of we only doing this trick to speed up our process because we don't want to predict this 18,000 targets, yes, here. But actually, you can see from this that it gives us improvement of the quality, because what we have dropped out, keeping only these components, probably was unpredictable noise. And when we drop it out, we get better results. Yes? Okay, so if we do some simple modeling with rich regression and just one feature, it's a compound, and one hot encoding with carefully selected by cross-validation parameter, we'll get this kind of score. With some optimization, we can get this kind of score. And then there is a trick which was proposed by the Song and the Luda, that if you make a blending of this solution by a simple model with more simple solutions, which are just the averages by the compound, that means this solution, and by cell type with these coefficients, you'll get this kind of score. And what as the public, other publics are doing, this are kind of combinations of this idea. So they a little bit change the model, not rich, but maybe something like that. And I think they're a little bit overfitting to the leaderboard, because they kind of, I don't see the cross-validation in this public notebooks. So that's maybe main ideas which you have on the public notebooks. And not public, but top places on the leaderboard are the following, as I said. Yesterday it was like this, and it was the same for several days, I think maybe for a week, and today there is a change. Yeah. So kind of important questions, I think, what, sorry. There is a question in the chat. What led us to such a big gap to what team employees at Novel to have that? No, it's private notebooks. I don't know exactly, but since it have been done quite in a few days, I don't think there is some non-trivial ideas. I think they just something like so what everybody is doing, looking on the public notebooks and just trying to blend them, yeah. And maybe you fine-tune the parameters of the models somehow, yeah. So I don't think this difference is quite substantial for the moment. Yes, it's a kind of beginning of the competition, and I think it will change. Yeah, in several days or 1 or 2 weeks, this will change. We will see better public notebooks, and we will see better top score results. So maybe we don't need to care much about this. Yeah. No, I'm not. Do you think blending and fine-tuning will be helpful in this competition with such tiny data set? Well, I think yes, but of course I cannot be completely sure. Yes, I have now fine-tuning by the cross-validation by the Ambros team. We can discuss it. I'll show you later. And it somehow worked. Yes, so for example, with fine-tuning, I think that when I improved cross-validation score, my leaderboard also improved. So I think some fine-tuning works. And we have, I'll discuss it, we have kind of infinite possibilities to fine-tune this kind of and blending, also blending, you see the blending surprisingly works. Also, that's a little bit strange for me, but you see that just people, what Song and Luda did, that they just blended this solution with more trivial ones, and the score improvement was quite not bad. That was unexpected for me, honestly, that in such a way from kind of these numbers or these numbers, you can get this. So, and the other post on the forums, the other people say that for them, blending seems to work. So I think it might work. But I agree, this is a little bit strange. So at the beginning, I didn't expect that blending or fine tuning will work very well. Yes, civil leaderboard correlation is a good sign. Yes, I agree, but it's not perfect. Yes, so for some cases, I see the correlation, but not for all of them. So no, again, very small data set, so we can expect everything can happen here. Any questions? Denise, any questions? Antonina? Yeah, and question, I have a question to all of you. So a funny question, and just don't be very serious. What do you think when we end the competition, what will be the top score on the leaderboard? Will it be something like, I don't know, I think we will finish somewhere at maybe like this. So colleagues, if you don't be shy, so just say, what do you think will be top score on the leaderboard? Just please send into the chat. Wow, you're very optimistic, Antonina. 0.45. I don't think this data is so much predictable. Okay, so 0.5. I think it's also optimistic. I'm not so optimistic about this. I think we will not go quite far. But colleagues, don't hesitate. Just send your guess. What do you think it will be? And in 2 months, we will see who was right. Arbitrary choice of weights for blending, such as can be a good. Yeah, sure. Yes. Yeah. You think 35 will be reached? Wow, you are very optimistic. Wow, let's look.
Yeah, I wonder, you, Alexander, you're working in the area of single cell sequencing, right? So you probably know what is the intrinsic noise for sort of pseudo bulk average for a single cell type, and relatively, I would say, like a major cell type, like.
Well, yeah, I'm working in single cell, but maybe this problem is kind of unusual for me. But my experience, maybe from the last year competition, that it was something like that, that there were some simple baselines with the reach, for example, and we have not go much far from these simple baselines. So I think this data is also very noisy, and situation will be the same. We created the simple baselines, and I don't think we'll go much far from them. That's my expectation. That's why I think it will be something like this finally. But people are more optimistic. I might be wrong. That's difficult to say because it's kind of everything can happen. Yeah. So yeah. Yeah, and so just. What I also don't understand that, for example, what scores will be valuable for some biological insights. Yes, if we get score, I don't know, 0.5, 0.4, what kind of biological insight will we have? That's, I think, would be nice to clarify from the organizers or other people in the field. Yeah, and what are the burning questions in the biology we can answer with this data? Yeah. So for me, it's quite interesting. I also don't know. Yeah. Okay, so another technical remark, the metric and the outlier. So the metric is very simple. It's just more or less the standard mean square, but it's calculated row-wise. So that means in the direction of the targets first calculated, and then averaged over the samples. As any mean square metric, it's sensitive to outliers, and we should be careful about that because there is a post on the forum by one of the participants that he says that there is an outlier, and in his point of view, that outlier kind of creates some problems. Yeah, So in particular, if we calculate something like cross-validation score, it will be much higher than we see on the leaderboard, and probably it's due to this outlier. So we need to be careful and pay attention on this. Yeah. For me, it's one of the points I should do, but I haven't yet done it yet. Okay, so let me say for those who are data scientists, send a bit of biological introduction. Yeah. So in the targets, what we have these symbols like A1, BG, or something like ZX, DA. What is it? It's the name of the gene. So if you go to Google, just put this A to M bar and then gene, you will see a lot of results. Typically, we know a lot about many of these genes, maybe not all of them, but many, yeah. And for many of the human genes, we have just a particular page on Wikipedia. For example, this GATA1 gene is quite important, and there is so about, I think, several, maybe 10,000 or maybe even more genes. We have separate pages on Wikipedia, and there is a lot of information. Yeah. So if you don't know what is it, just try Google Wikipedia and another resource, which I'll see you just in a moment. So, so how to think about the genes and what are the, so let us think about the cells like humans. So humans need to eat, move, breathe, proliferate, communicate with each other. The same is about the cells. So they do similar things. They need to kind of eat, move, breathe, proliferate, so on and so forth. And the different genes, they are responsible for different functions. So that's why kind of we have so many genes, because cells needs to do many things. Yeah. And many of these functions are known to the science. The problem is that one gene actually can do several things. And this is already less known, yes? And so I think it's an interesting question. Can we incorporate the biological knowledge, and how to incorporate it? It's one of the questions in the judge prizes. I'm not very optimistic. I don't think that there is an easy way to, or there is any way to incorporate this biological knowledge, but it's interesting to try. But for those who are from data science, I think it's good, because that maybe biologists do not have a competitive advantage. Yeah. Another resource which I wanted to mention about the genes, this is a so-called human protein atlas. What is kind of maybe fascinating, that this resource partly was created with the help of the Kagglers. So there was competitions on the Kaggle Human Protein Atlas, which was some about image processing, and so there was something, I think it was several years ago, and the insights which Kagglers provided were really used by the Human Protein Atlas team. I have heard the talk by Emma Lundberg, who is one of the organizers of this project, and she said that indeed the solutions proposed by Best Fitting, very well-known grandmaster, for example, were very useful. Yes. So if you are from data science point of view, this you can go to this, and you don't know something about the biology, some genes or some cell types, you can go to this resource, just Google Human Protein Atlas, for example, T cells, and it will give you quite a lot of information. For example, it can be useful for T cells. We have these genes which are particularly expressed. Yeah. So, or for example, this is from Wikipedia. And just also might be important picture that for the genes, they have such a kind of pattern. You should go to the RNA expression and press here, show, and then it will show you in what cell types this gene is particularly activated. You can see that some of the genes, they are active in some cell types, but not in the others. That might be important for our competition, because we have different cell types, B cells, T cells, and might be some genes activated only in, for example, B cells. Yes. And maybe we can take this into account somehow. Maybe not, I don't know. okay, maybe this is more something about the biology, that the genes, so what should we keep in mind, that these genes are very correlated. So the fact that we have reduced dimensions to 20 or 35, and then take the full matrix by inverse transform and obtain good results, it's not very surprising, because genes are very, very correlated. And if it's correlated, then this PCA 25 components, it's a kind of takes the main signal from the matrix. And so that's why it's maybe not surprising. So here are some examples of how the genes can be correlated. For example, this they can join into the complex of the proteins. This gene is transcription factor, which activates many other genes. This is a kinase which phosphorylates this protein. So there are many, many relations. Yeah, and in typical, so the genes are very correlated. Yeah, okay. So now let's go maybe into more details of the data science approaches. So so we have categorical features. How do we encode them? So we can so the simplest way is one-hot encoding. Yes, just one hot. The more sophisticated way is target encoding. We can use also this Hembert shared by Alexey Trepetsky, and we can use molecular descriptors for the compounds, which Antonina, I hope, will present to us today. Yeah, and it can be, for example, ordinal encoders. So this topic of the categorical feature encoding is quite popular in data science. There were, for example, 2 competitions on the Kaggle, which just were devoted to the working with only categorical features. Actually, there was some difference because it was binary classification. And now we have a regression task. But still, I think many approaches from this, we have lots of notebooks shared in this competition, and they can be useful. Actually, there were 2. Categorical feature encoding challenge 2 and challenge 1. Yeah, so we can look on this data set. Also, there was a benchmark written in 2018, 19 by Denis Vorotentsov. It's a quite useful and quite well-known paper. And there is another benchmark recently published in News. So here are the links. Everybody can look for that. Yeah. So I think the most useful is the so-called target encoding, and it's useful to use so-called smoothing. So target encoding, what does it do? It takes category, and you just calculate the mean over target for the samples across that category. So that means we have many samples for which this feature equal to the one of the categories. So we fix the category, and then we just calculate the average of the target, and we do it for each category. And so from the targets, we encode, we get the numbers for the category. So this is encoding, yeah. And what is important that this encoding has a parameter called smoothing. Smoothing makes a kind of average between the simple encoding and the mean over all the samples. So it creates something like a bias, yeah. So we average 2 things. If you put this smoothing very high, then what you get that this part will be almost neglected, and you will see this part which doesn't depend on the category. That means this feature will be just a constant. Yes, and so, and if feature is a constant, the model cannot use it very well. So if we think the model, the feature is quite noisy, it's better to make it constant or something near to the constant, such that model will not use this noisy feature. And what people do, they just select this parameter by cross-validation. Yes, so you can try. We can. I have tried this, and for simple models it worked. Any questions, comments, suggestions? Okay. Now, just one more remark. So there is a library, Python library called category encoders. And you see there are many encoders. So I think this target encoder, which discussed on the previous slide, is the most important. And it's important to tune by the cross-validation this smoothing parameter, but we have much more. And we can try all of them. Actually, we have a way to generate feature engineering, lots of many unlimited number of features, because we have many targets, yes, and we can make target encoding for each of them. Or we can take PCA, I say UMAP reductions of these targets and make encoding for these reduced targets. Or we can make some combinations, for example, biologically kind of motivated. We can know, for example, some pathway or something like, then you calculate the average of the genes for this pathway and make a target encoding with this combination, with this signature. So there is practically infinite number of the opportunities. But I'm not very optimistic that this can achieve quite good results, but still we can try. Yes, so I think the most useful is target encoding with smoothing the parameters. Yeah, but yeah, and target encoding applied to the PCA or TSVD components. Is it clear? Antonin, is it clear?
Yes, I wanted to ask how you how to look for the best smooth parameter by cross-validation. What's the process?
Just we said, for example, I want this smooth parameter, some grid of the values, for example, 1, 10, 100, 1,000, so on. Calculate the model with each value of the this smoothing parameter, and just choose the best one.
I see. Okay.
The best for the I mean in cross-validation score. So
have,
this is,
at the start, you have a vector of some numbers like 001 or 001, etc., right? And try all of them.
no. So this parameter smoothing, yes, it's one number. Yes, for one feature, it's one number. And so you can just, as I say, take a grid, something like 1, 10, 100, 1,000, 10,000, so on, for this parameter. Calculate the cross-validation score for each of these values of the parameter. And then choose the best one. Not clear?
Yes, I see. Thank you.
Okay, so yeah. So another way to encode our features is this ChemBERT, which is BERT is a popular natural language processing tool, and what we can there was its modification called ChemBERT, that means chemistry BERT, so it can work on the smiles. And it had been trained on 77 million of different smiles, and considered not now it's a kind of quite good in this research community of the chemicists, computational chemicists. So Alexei Trepetsky kindly shared this data set and notebook. So just in his data set, you have already encoded by the ChemBERT all these molecules. They provide some kind of embeddings, vectors. And so we can try to use these embeddings as a features. Yes, instead of target encoding, or together with target encoders, or together with one-hot encoders. Yes, and it's one of the interesting ways. And one of the ways which I think is quite interesting is to fine-tune this ChemBERT. So as far as it can be possible to fine-tune it to the particular task, as we do in natural language processing. And we can try to do this for this particular task. Yeah, I am not sure how successful it can be, but it's interesting to try. Yeah, and one of the things which I want to mention, it is a hard way. That means, so what we have discussed so far, that we have this small number of samples, and we are trying to predict the 255 samples, which are pseudo bulk. So we are trying to predict for not for the single cell, but for these averages of the cell types. But what we can do, and it was commented yesterday by the organizers that it will be interesting to do from the research point of view. We can first train the model on the single cell data and then make a pseudo bulk and calculate p-values by lemma. Yes. In this way, we have lots of data. So now we suffer from the problem that we have not so many data, only 600 samples. But if you look on the level of the single cells, we have 200,000 cells. I have looked in their file. Yes. So the number of samples is incredibly bigger. But the problem is that noise for the cells also quite bigger. So when we calculated this pseudo bulk, we reduced noise substantially. It's not clear whether this approach can work. There are many research papers which try to predict, and they report that they predict very good, but on the level on single cell perturbations. But there have been no let's say, independent benchmarking of all these papers. And this competition is a kind of the competition which can benchmark if these papers really work very well, so we can try to apply these methods and get a better score. But that is, I'm not very optimistic about that. Also, I'm afraid of these technical difficulties, yes, because of this, we need to calculate pseudo bulk and then lemma, and all these bioinformatic packages, they have small kind of details which you need to tune, and if you are not an expert, I'm afraid that...
Do we have the code, the procedure that we use to calculate the regional values with lemma?
Short answer, from yesterday, yes. But so the organizers promised to share this code from the beginning, but it took them 2 weeks to polish the code and share that. So that's clearly this not so simple. Yes, that's why one of the reasons that I'm afraid of it. But yes, the code exists. Yes. And if someone is an expert in lemma, for him, I think it will not be a problem. Yes, just for those people like me who just not working with this lemma every day, I'm afraid to dig in this. Yeah, but yes, I think it's very interesting approach, but it's not clear. Also, if we can do this, we can answer another question proposed by the organizers, which is important for this judge prizes about this ATAC data. Yes, we can do the model using these single cells, both on RNA and ATAC data, and then try to predict, do the same thing. Yes. It is very interesting, but it's a kind of complicated, yes, approach. It's complicated and not clear, kind of, is it promising or not? That's a problem. Denis, maybe you have some comments, I don't know, for this. What do you think? It will kind of, should we spend time on this or not?
I honestly don't know, because I don't know, like before that, I got some internet issues, but I wanted to ask you, what is your sense, what is like the level of intrinsic noise for those type of data? Obviously, like it's much more complex, because here it is a differential signal and it is even log p-values. But if for a second we will forget about log p-values, and instead we will assume that we are predicting raw expression values counts, then what's the level of noise between, let's say, 2 donors with a single cell type with this sort of broad cell type, I assume that it can be quite significant, right? Even if the procedure is exactly the same.
Yeah, I don't know how much about the donors. Yes, I think one of the main things is this dropout, that we have these zeros. Yeah, that kind of for
sure.
the single cell is that we should have expression for some gene, but we have zero, and we know that it's kind of technical, but it's kind of it's. You need to do something with that. It should be, for example, 10 or 100, but you see zero. It's this dropout can be very big. Yeah, just because it's a random event, which was kind of after this.
Yeah. So it would be interesting just to sort of look at the variant structure in the original data, because we have 3 donors, multiple plates, multiple perturbations, a lot of cells. Then we can take a look at the variation between a single donor, like, and cells falling into a single single cell line, and if the variation there will be sort of acceptable, will be relatively low. Like, I mean, the variation for a single gene inside a single gene compared to variation across genes, right? Then I guess it makes sense. So I am definitely much more interested in the second approach, and if I will be doing this competition, I will be trying to do it on single cells. But I am not sure that this will be what will win eventually. Maybe not.
Yeah,
but in general, my interest here is to try generative modeling for single cell expression.
Yeah, it's interesting approach, but I'm not very optimistic, but maybe it's just because I am not optimistic. You know, I don't know. Yeah, but I had some talks on the generative approach for single cells, and it was several years ago. They were not convincing for me, but maybe situation changes. Yes. Yeah, and these are the pictures on the level of the single cells. Yes, so this is the standard UMAP. That means each dot here is a cell, and you see some clusters. I haven't yet analyzed what are these clusters. I think some of this is some cell type. It's probably T cells or B cells, I think, and these are the other, because it's a marker of the cells. Yeah, and so also I plotted these pictures with the cell cycle, which I like. So I see that there are some proliferating cells which are here, and this main part is not proliferating. Yeah. So Denis, maybe answering you a little bit in the direction of your question. So ideally, this picture should be a triangle like this. It should be just like. But you see what we have. And this
so
is
have they
not for one gene. This each coordinate G1S calculated as average of 50 gene. So you can imagine that if you have such a kind of noise on average of 50 genes, if you look on one particular gene, and these genes are selected, they are kind of very strongly expressed. Yeah. So if you look on some genes which are low expressed, the noise can be very, very big. It depends on the genes. Yes, of course, some genes are very expressed, and then noise is not a big problem. But
and regarding the code used to obtain those differential expression values, it contains the whole pipeline, right? From preprocessing steps on single cell data down to differential expression on bulk data. Or actually, I don't know, maybe they even do differential expression on single cell and then aggregated.
Yesterday there was meeting, or 2 days ago, meeting with organizers. It's on the YouTube, and they showed this code, but they didn't discuss it in very details. But as far as I understand, yes, it is the whole pipeline. Yes, but
so I just cannot see it actually on the site, on the Kaggle page. Maybe I'm
It's not
just
on the Kaggle. They shared it on the Saturn cloud. If you go to the Kaggle and discussion, one of the first discussions is that you can use Saturn cloud, something like that. It's a pinned by the organizers. It's on top.
yeah, I see. Okay.
And you should
okay,
go to not to the Kaggle, but to this powerful Saturn cloud. And the code is there because they say that you cannot run it on Kaggle because Kaggle gives you not so much resources. It's you need more memories. And
I see. I was actually going to run it on my own computer, but one last question. So I still don't quite understand how the test set looks like. So yes, here I see that they have hold out information about certain cell line, or I don't know if it maybe cannot be called cell line, but like cellular plate experiments.
cell types.
Yes, cell type experiments. But is the number of donors the same? So it's still the same 3 donors, or?
As far as I understand, yes. As far as I understand, everywhere, all these 3 donors are averaged. Each box is as far, but this may be better to confirm from the organizers. But as far as I understand,
Okay.
But yeah, it's better to confirm. The very good question. Yeah.
And,
Just one remark that you see here, light blue and not so light blue. This is private and public data set. Just a remark. Yeah.
And the cell types, were they determined by surface cell markers or by analyzing the expression itself?
I think by expression of the cells. So in this data, comparing to the last year, they do not have surface protein markers, only the RNA transcriptome and ATAC.
Interesting. Wait, they also have ATAC?
Yes. Yes. So this, and one of the questions for which they can give you a research prize is to take this ATAC into the account.
Interesting.
They emphasize that it's important. Yes. So how did you integrate the ATAC data? Yes.
Yeah, it's probably this multi-ohm OBS, multi-ohm packet files. Okay.
Yeah.
Yeah, interesting. Yeah, that's kind of cool.
Okay, so my part is finished. Antonina, do you have time to present today, or what's?
Yes, I can. If you still ready to listen.
Okay, let's try. So Antonina has a small short presentation about these molecular descriptors, and she shared the notebooks on Kaggle, and also now she can explain us these molecular descriptors. So I stop sharing for the moment, and now I allow you to share, and please start.
I have to see the button to share screen. Yeah.
Should be green. It's distinguished by green color. Yeah.
Do you see it?
It's coming. Yeah, now I see it. Yeah.
Okay, so this is a really short introduction for a little bit mostly manual work of extracting molecular descriptors. Actually, I would divide all the techniques to get features for our drugs and cell types by two different kinds. First one is like target encoding is grouping of drugs by their effect on cells, right? And you also can group drugs by their chemical structure, which is molecular descriptors do. And here I just extracted all the available descriptors from the package in our special package here. And there are 53 of them, a lot. And the big challenge is how to select relevant ones. It's not trivial task, actually. So each descriptor represents some kind of information about the drug, maybe structural, maybe physical, chemical, or anything else related to the chemical structure of this compound. And each descriptor has several parameters also. So it's much more data than 53 features. It's much more. So as a little breakdown of workflow here, I just extracted all available. And then I removed these which produce errors or warnings, because they need some more information than we have in our smiles. In our data set, we have smiles, right? As Alexandra mentioned, which represent two-dimensional chemical structure. So from them, you can get some information about the molecule. And these descriptors highlighted in red are not extractable because they need some more information than we have in smiles, like 3D, something like this, or should belong to some kind of molecules, specific kinds. We don't have them, so I just removed these descriptors. And remained 44. And also, I'll see the code here. I'll show you the code here because this is in this public notebook, feature engineering. You can see it. So these are all 53. And these warnings I said, molecule must have 3D coordinates. We don't have them, so we cannot use these descriptors. I removed them, and after this, I retract the information, this code. Anyway, I've got some warnings, which may be quite important, because they say that two molecules, this one, you see the name, delanzamib, and this one, MLN2238, get NA for two descriptors, this one and autocorrelation charge. Two descriptors produce NA for two molecules. This can mean that these molecules are some kind of outliers based on chemical structure, which I did not yet examine more precisely. So you may want to check it afterwards. So to not have NA in our features, I also removed these two descriptors. So we come up with 42, just occasionally. Also, I did some more analysis here and removed constant features and columns with NA. I said already about NA, and constant features make meaning that some, I see the table. Sorry, I've lost in my notebook. Some descriptors produce same values, meaning that not differentiate compounds from each other. So not valuable, not useful for us. So also removed here. And we come up with 167 parameters. I say parameters, not descriptors, because each descriptor is represented by several parameters. It may be like small ring. It includes about 7 parameters. You get not one value, but 7 values, vector values. And each descriptor have from 1 to maybe 33 different parameters, and so that we have from 42 descriptors 167 features. After all removals, the remaining descriptors here, and the main work here was to try to classify them somehow and get some understanding of what does it mean, what does it say about our molecules, and how they are relevant to this competition, to group our drugs, to code them. And here you can see this was my manual work, and I'm not quite sure, it doesn't have some mistakes, but I guess quite correct. I divided it for 3 groups: structural features, those about physico-chemical properties, and about molecular complexity. And here are 3 tables for each group. Here I added some information about the descriptor and some more info about summary effect. Like this one is about overall complexity of the molecule. The second one is about 3D shape of this molecule. This one about branching and I don't see it. Zoom. And connectivity, and etc. I will not read all of this. I will share this presentation, and you can read by yourself afterwards. So, second group is structural features, and there are more. They characterize our drugs like amount of aromatic system, branching, conformational flexibility. Actually, to get some knowledge on how to choose, even having this data, this information, you have to have a little bit of chemical background or some time to Google it and get some understanding. So, and third big group is molecular descriptors that characterize physico-chemical properties of these drugs, like atomic mass, charge distribution, polarizability, and lipophilicity, etc. As you can see, all these characteristics may be valuable because they can somehow be related to the ability of a drug to affect gene expression, like lipophilicity. The more lipophilic the drug, the more the chances get into the cell and affect it. Kind of this thinking you can apply. Solubility, permeability also. So I think this one quite important, as well as structural features also, because the more functional group, for example, the drug have, not the more, actually, different functional groups may cause the drug produce different effects. So it's maybe also important descriptor. And finally, after you get some knowledge about any one, these 3 groups, it's useful to finally select from 167 molecular features, the most important and relevant. And you may want to apply some techniques to feature selection, because if we just plot a heat map of correlation matrix between all these features, we see that most of them are highly correlated. So they cannot be used as they is for now, but need some educated guess and selection process. I think you may have some questions. I don't know.
I have some remark, Antonin. You mentioned this MLN 238 as an exception, that it very far from the others. Yes, there was
Sorry,
some drug.
To drug?
Yeah, two
Yeah.
drugs, and one of them was MLN something. Yes.
Yeah, I saw this drug also in the discussion, I think, also
it-
producing outline effect
Yeah,
on the
yeah.
genes. Yeah, that's why I said it's important because already we have 2 poems. This is an outlier also based on its effect, as somebody mentioned in the discussion, and also based on its molecular structure too, because
Yeah.
yeah.
So
And this one
what
also.
does it mean? Cat has n. So do we understand why NA for this discrete? What
Yeah, to get some knowledge of this, I didn't have much time actually. I just made it yesterday. So this analysis need
Okay.
somebody to deep more down. So you can
Okay.
Google this molecule descriptor and see what it does, what does it mean, what it
Okay,
calculates
okay.
exactly, and you will know what this molecule do not have to
Yeah.
calculate this stuff
Okay.
for it.
Okay.
Yeah, but
Have you tried-
100%, yeah, we can say already is that these 2 are outliers based on chemical structure.
Hmm. Okay. Yeah, very interesting. Thank you. Have you tried some model to submit prediction based on these features?
Not yet, because I didn't do this job about selection. It's not quite easy.
But you can just make a PCA from this and no need for selection.
Maybe, but I see from the descriptions there are like duplicates, like this one, eccentric complex name about connectivity, and there are 4 others doing the same job. So I think you don't need all of them. You have to select one.
I think if you do PCA, it will kind of take care of this automatic. No, not very well, but somehow. So anyway, I mean, I think.
Yeah, one question. Those descriptors, are they mostly represented by floating points, or are they integer values? I see that there are at least some integer values. So in this sense, PCA may not, it may be okay, of course, but it also can be more sensitive to those correlations. But anyway, it's not really that much of a problem for
yeah, you should.
machine learning approaches.
Yeah, you should look more on this data in the output. Here you can find this table. Let me mole descriptors features this one already ready for use. And here you can examine the distributions of the data, and you can see double, and integers, and also zeros and ones. There are binary features, like amount of something, like maybe aromatic rings or something. That molecules can have only 0 or 1, and there are a few of them here. So maybe you have to filter out based on many zeros.
Yeah, okay. Yeah.
So my thinking of this is chemical structure is really important, and we should check this. I maybe will check in my public baseline and share the results there.
And so in your notebook, you have this Morgan fingerprints Tanimoto. It's different from the chemical descriptor.
Yes, I said in the beginning, you can get a lot of information from chemical structure, not only descriptors. The other thing you can get is Morgan any fingerprints. Actually, Morgan is just one kind of fingerprints you can get from smiles. The basics is here. You can read it after if you want. There are some links, I think.
It's some features. I mean, this is some numbers, or this is
Fingerprints produce binary matrix. Here is a 1025 columns and all zeros and ones, binary matrix. It also represents chemical structure, but in other way than molecular descriptors do.
okay.
Also
But,
here you can.
So we can again, and have you tried to submit this feature model on that?
I tried fingerprints, but they do much worse than target encoding. But I didn't do anything. Just use as a is zeros and ones binary. Maybe that's the thing. You may have to do some, I don't know, dimensionality reduction here also.
Yeah, I think it's first thing to try is some PCA, something to maybe 5-10 components, something. There is a question: can these fingerprints be used as embedding for molecular, or will they have similar chemical? Is nearly similar fingerprint? Will they have similar? No, I mean, so the question is: will this if the fingerprints are close, does it mean they have a chemical properties similar?
Yes, but it's different for each descriptor. For example, I see here. For example, like where it was, like molecular weight, right? It will be different for each compound, and I don't know how well molecular weight will characterize our compounds about their action on gene expression, maybe not so well. And another one, like this log P, it characterizes lipophilicity, and it sounds quite much better than just molecular weight, and will differentiate the compounds better, I guess, but it's my guess.
Yeah, okay.
So each one can produce I think maybe a good way to visualize it somehow by plotting PC of DE values from targets, right? And color by each of these descriptor, and see which one do good grouping, clustering, right?
Yeah,
it would mean
we can try. Yes, many of them. So, yeah, and if there is, I think it's more simple to put them into the models and just check the score of the models.
Yeah, for very for good start to check just it. But actually,
okay. Yes, sorry for interrupting. Actually, my question was a bit off topic. I just wanted to ask if the descriptors, if RCD-kit that you RCD-K, I think that we used, if it is similar to RD-kit on Python, and if Python has the same set of descriptors implemented.
Yeah, the link is here. You can use Python and library and packages.
Okay, thank you.
This link. And the description is mostly from there, actually, from this package.
Okay, great. Because I see that CDK, it seems like it's maybe even neither Python nor R, and then it has APIs on top of it with R and Python interfaces. Maybe it's not a fact. I need to check.
Yeah, there may be some difference. Actually, I didn't find documentation, detailed documentation about how R do this, and I used Python, so I hope this the same, but maybe not. But should be close, at least.
So what's your plan? When you think you can submit the models with these features and kind of share with us your results?
Yeah, I will.
Okay, okay, great. Okay, thank you very much.
Yes, thank you very much. Thank you, Alexander, as well.
Thank you everybody for participating. So any questions, comments, suggestions, and if someone wants to share something or maybe kind of participate in some discussions, please don't hesitate to kind of notify me or other colleagues. So I think we can kind of make more kind of meetings, maybe more with discussions, yeah, maybe less talks. So today a kind of introductory talk, but maybe then we can make more discussions. Anyway, so everybody is welcome. Don't hesitate. It's kind of very informal. Okay, so thank you very much again, Antonina and everybody. And maybe then no questions. Okay, then good evening to everybody.
Thank you.
Thank you.
Goodbye.